Okay, so this is going to be the CSEC Biology crash course. So basically, we're going to be going through a portion of the syllabus, and basically, we're going to just review some of the main parts or the main stuff in the syllabus. So, yep, you can go ahead and subscribe. They got the Instagram, the playlist, the textbooks, all that stuff. And without further ado, let's begin. And then right here, just some motivation. So basically right here, it's just a Bible verse that says I can do all things through Christ who strengthens me. So we can just keep that in mind as we get started. Okay, so now some of the main characteristics of life. So basically, when we're talking about biology, we have to start right here, because if we don't start right here, we cannot explain anything else that come afterwards. So basically, we know that biology is the study of living organisms and obviously their interactions with the environment. So basically, how they grow, how they respire, their sensitivity, their movement, nutrition, excretion, reproduction. So basically, in order for something to be considered living, it has to perform all 7 of these functions, right? So if you ever see an organism and you wonder if this organism is considered living or non-living, you have to ask yourself first, does it grow? Does it respire? Is it sensitive to stimuli? Can it move? Its nutrition slash feeding? Is there any form of excretion? Is there any way to get rid of waste? And then obviously reproduction to produce more offspring. So for example, there are many different types of organisms, anything ranging from a tree to a tree which is a plant to animals. There are different mechanisms that each organism uses. So for example, the way how a tree grows is not like how an animal grows, because obviously with animals they grow to a certain height. For example, human beings, we grow to like a certain height, probably not past 7 feet. I don't know if there's anybody taller than 7 feet, but you get the point. The way how we get rid of waste, we know how humans get rid of waste and versus animals, because we're technically the higher class of animals, but versus how a plant gets rid of waste. So there are many different variations in it. So you just need to take into consideration these 7 characteristics of a life, because this is what makes us living. So we're going to start with the simple stuff, right? We're going to start with the simple stuff. So right here we're looking at an animal cell, right? But before we really talk about animal cell, I want to mention something else before this. So let's talk about prokaryotes and eukaryotes, right? So just to jog your memory, we know that prokaryotes, those are basically our bacteria cell. They do not have a true nucleus. They basically they are very small, they are unicellular. We know that about our prokaryotes, right? And then we have our eukaryotes, which is where plants and animals and all that stuff fall into, right? So prokaryotes, it's a very big it's a very big group with just kind of like one thing in it, right? And that is bacteria, right? So that's what prokaryotes is basically. So it would fall under the category of bacteria. So bacteria, prokaryotes, same thing. And then eukaryotes, our plants and animals would fall under that category, or fungus under that category. So just bear that in mind, that we need to be able to differentiate between a eukaryote and a prokaryote, and just know that prokaryote means before a nucleus and then eukaryote means through nucleus. So this animal cell that we're looking at would fall under which category? It would be a eukaryote, right? Because we do have a nucleus. So let's look on this animal cell, and this is very, very important if you study biology, and especially for CSEC. Please know how to draw this, because if you're going to do CSEC biology and you really and truly cannot draw an animal cell, I don't know where you do at all, because this is the simple. This is basically the simple of the simple, right? So your entire biology life or career, maybe you don't plan to study any more biology after this, but you need to know how to draw this. You cannot say you did do biology in a school and you don't know how a little animal cell look, right? And you must be able to reproduce this drawing if needed, right? But yeah, so it does help to be able to know how to draw this. So basically, our animal cell. So we're going to take a look at it. So the first thing when we look at this animal cell and we're looking at the shape, we can see that it has a very irregular shape, right? Very irregular. It's not fixed, right? We do not have a cell wall. We absolutely do not have a cell wall. It's not fixed. It's irregular, right? It's not fixed. No cell wall for our animal cell, right? So let's take a look inside, and obviously let's talk about the cell membrane. So we don't have a cell wall, but we have a cell membrane, and obviously the cell membrane controls what moves in and out of the cell. So you can just bear that in mind if you ever see an exam or even an MCQ, maybe on the paper one. Just know that it controls what substances move in and out of the cell. For example, nutrients, ions, oxygen, things like that moves in and out of the cell. And even get waste out of the cell, it have to pass through the cell membrane. Now we're saying that right here, our nucleus, right here. So it's our nucleus, and inside is our nucleolus. So basically, your nucleolus, it produces ribosomes, and our nucleus right here is basically what controls the cell. It is what controls all the functions. It controls all the cell organelles. And now we bring up the word cell organelles. So all of these parts of the cell are called cell organelles, or nucleus, or cell membrane. All that stuff, they are cell organelles, right? And then outside of our nucleus, this is our endoplasmic reticulum, right? So this is our endoplasmic reticulum. Now we have two endoplasmic reticulum within the cell. We have our smooth and we have our rough endoplasmic reticulum. If you need a way to differentiate it, just pay attention to the words. When you say rough endoplasmic reticulum, we're talking about ribosomes. You can see the ribosomes on it, on the rough endoplasmic reticulum, right? And that is because our rough endoplasmic reticulum makes ribosomes, right? So these ribosomes help to make protein. So that's why it's there. And do also bear in mind that we also do have ribosomes scattered inside of the animal cell also. But just bear that in mind. And then our smooth endoplasmic reticulum, it is basically smooth, absolutely no ribosomes in it basically. So the smooth endoplasmic reticulum aids in basically lipid and steroid production. You can just keep that as a definition, as a function. Then our mitochondria, which from probably even grade 7, yeah, I hear that this mitochondria is basically the powerhouse of the cell, right? This is the energy bank of the cell. It is what supplies the energy for the cell. We have our vacuole, and basically it serves as somewhat of a storage organ, kind of like it stores waste and also water, right? So the vacuole is what would get rid of the waste and store water as well. Then our Golgi apparatus, it has these stacks in it. You can see that it's very much stacked up together. So this is our Golgi apparatus, and it is basically the packaging center of the cell. And then obviously ribosomes, these are free floating ribosomes in our animal cell. These also make protein. And then our cytoplasm, which is basically the jelly-like fluid that holds all the cell organelles in it. And then we have our lysosomes, and it basically is its function in the cell is to basically break down stuff into smaller parts, right? And then this centrosome, you don't necessarily have to worry about this. This is mainly involved in cell division. So at this moment, you don't have to necessarily worry about the centrosome. We can talk more about this when we're talking about cell division. So for now, we don't really, but just a brief as to what the centrosome does during cell division. It's gonna separate to the opposite end of the cell. Spindle fibers are gonna extend from it. Chromosomes line up, pull, then it's gonna pull back, all that stuff we get. But we're not getting into that too much right now. But that's what the centrosome would basically do. So then we have the animals, not the animal, the plant cell. So this is our plant cell, and we can see differences between the animal and the plant cell. But the same thing that we did before with the animal cell, where we need to classify this cell, right? So plant cells are also eukaryotic cells. They also have a true nucleus. So basically, the structure of a plant cell is very, very different from the structure of a animal cell, because the first thing we notice is that cell wall, a fixed rectangular cell wall around it. And if we look back at our animal cell, that was not there before. So basically, the cell wall, which it mainly provides a rigid structure, like a rigid protection barrier for the cell. It prevents the plant cell from bursting. And the reason why it's so important, especially for a plant cell, is because plant cells take in a lot of water, right? They take in a lot of water, and you can see that they have this huge vacuole in the middle, which we're going to talk about very, very soon. But yes, plant cells take in a lot of water, and they need to be able to maintain the cell so that the cell does not burst. Right, so that's the main function of having that cell wall, as a barrier, as a rigid structure, providing structural support for the cell. Because we know that plants, for example, trees, they need a lot of water in order to grow. They absorb a lot of water through their roots, and so this is why plant cells need that rigid structure, which is the cell wall, in order to prevent it from bursting. So yeah, then we have the nucleus, which serves basically the same function as in the animal cell. As a matter of fact, the nucleus serves the same function right across the board, controls activities of the cell. The cytoplasm, still that jelly-like substance that holds the cell organelles and all the contents of the cell. And then our cell membrane, which the cell membrane was also in the animal cell, and it's also here in the plant cell. Okay, so you can basically say double the protection for the plant cell. But basically, this cell membrane is permeable, just like in the animal cell. Substances can move in, substances can move out as necessary by the cell. And then our mitochondria, very, very important. Still the powerhouse, still supplies energy for the cell, and it's basically scattered all around the cell, because you know that the plant carries out a lot of activities. And then we have our chloroplast. So basically, the cell wall and the chloroplast, and also the vacuole in the plant cell, are three things that we need to take note of specifically. We already explained the cell wall and why it's needed, right? Because plants absorb a lot of water and they need that structural support. Now, why do you think this plant cell have such a big vacuole that basically take up 3/4 of the cell? Why it needs such a big vacuole? For the same reason. The same reason that the plants absorb a lot of water. That's the main reason. And we said it before that vacuoles manage waste, and it manages waste, excretes waste from the cell, and also it absorbs water and stuff like that, right? So because plants absorb such a large volume of water, we need a larger vacuole in order to manage it, right? So that's the purpose. And then did this, so the yes, so in the animal cell, these vacuoles were very much scattered, but as you can see, they were significantly smaller versus the plant cell that just has a big vacuole in the center. So anyways, now onto the chloroplast. So this chloroplast is very specific. It's 100% specific to plant cells, or any cell that is able, or any cell that is able to carry out photosynthesis. Let me just say that. So chloroplasts, they have a green pigment inside called chlorophyll, and that is what is needed in order to absorb energy from sunlight in order to drive photosynthesis, basically, right? So that is why these small, those structures, those green structures called chloroplasts are necessary for our plant cells. So that's really the function of it. Alright, so now let's talk specifically about our bacteria cell, aka our prokaryotic cell. So we can see a lot going on with this diagram right here. A lot. So this is our bacteria cell. And the first thing that we need to break down is that prokaryote, it being a prokaryotic cell, it means before nucleus. So right and from the get-go, we do not notice this cell having a nucleus. There is no distinct structure in it that says this is a nucleus so far upon first glance. So we just go through that and explain what's happening right here. So the first thing that we want to start with is you can see that this cell has a capsule, right? This cell has a capsule, and by the way, prokaryotes are very abundant on the earth. Let me just say that, as in we have more bacteria and we have more, aka prokaryotes, on this earth than even eukaryotic cells. Just to put that out there. So we have a lot more of them. But the first thing, let's talk about the capsule. This bacteria cell has a capsule. Then it has a cell wall, and then it has a cell membrane, right? But so basically the plasma membrane is the cell membrane right here. So why you think this bacteria cell will need three whole layers? The cell membrane serves the same function as in plants and animals, controls what moves in and out of the cell, is permeable. The cell wall also provides that rigid structure, and basically the cell wall in our bacteria is made up of a polysaccharide. I think it's a polysaccharide called peptidoglycan. Me pronounce it peptidoglycan, but that's why I pronounce it. And in our plant cell from before, the cell wall is made up of this cell wall is made up of cellulose. This one is made up of peptidoglycan, at least that's what I call it, right? So same function, provides that rigid structural support for the cell. So now the capsule. Why do you think the bacteria cell has a whole capsule? So the whole function of having a capsule on a bacteria cell is basically for resistance, right? Now, as far as we all know, bacteria is not necessarily our friends, right? We obviously have bacteria everywhere, bacteria in our hair, in our mouth, our skin, right? But for the most part, most people view bacteria as very much harmful. So basically, the whole function of having a capsule is for resistance, right? So basically, it's to protect the bacteria cell. And that is why, for example, when someone is sick, for example, them sick and them say, oh, it's a bacterial infection. That is why bacteria is so hard to get rid of, because once you get it, the harmful one, once you get it on you, it's just the way it's designed. It's designed to be resilient, right? It's not easy for just dead out just like that. And partially, or mostly, it's because of the capsule. So the capsule protects the bacterial cell, allows it to withstand different conditions and basically be able to survive, right? So it is what promotes the diseasing, the permeable diseasing. It promotes the disease ability for the bacterial cell. And ribosomes, same function, they produce protein. Cytoplasm, same function as before. It basically holds the cell contents. And now on here we have these pili. So one is, okay, how we gonna, okay, so we have these pili, right? So we have the pili on it, aka pilus, plural. Or is it the other way around? I go again. I think it is pilus. Yeah, so pilus would be singular and pili would be plural. I feel like I saw it go, so just stick with that. But anyways, the interior function of these pilus is basically of the pilus here is basically to help the cell to attach to other surfaces. So basically it goes back to the nature of a bacterial cell. As far as we know, we are bacteria not necessarily friend, and we know that bacteria causes disease, disease causes illness, illness can kill you, right? So it's just the entire mechanism of how the bacterial cell is made. It's designed to be resilient. So these short here like appendages, they allow the bacterial cell to attach to different surfaces. And then we have this flagella. So the flagellum right here, this flagellum, and flagellum is singular, flagella is plural, but basically it's to help it move. So flagella is like a long whip-like appendage, and basically it facilitates movement of the bacterial cell, right? So yeah. Now, the main event of this cell is the fact that it does not have a nucleus, right? That is why we don't consider it to be a eukaryote. It falls under a prokaryote. So within this bacterial cell, what we're going to find is actually a nucleoid, right? And a nucleoid is basically the bacteria's DNA. So it's not enclosed in the nucleus like before in the plant and animal cell. The DNA is literally free flowing, right? It's literally free flowing, and basically this is the cell's genetic information. This is the DNA, aka the nucleoid, that's free, not enclosed in the bacterial cell. Like that's it. You can literally see it right there. You can literally see it right there. That is your nucleoid. So it does not have a nucleus. It does not have its genetic information enclosed. It is just open, free, flowing. And then the plasmid right here, the plasmid is basically just a circular, it's kind of like a circular DNA molecule, and basically it helps to carry some genes for the bacterial cell. So basically that's the function. Alright, so now we're just going to go through quickly the similarities and the differences between the animal, the plant, and the bacterial cell. So basically all three of them have a cell membrane, so we know that in common, right? And then all of them have cytoplasm, all of them. Then we're going on to the nucleus, where the bacterial cell is the only one that does not have a nucleus. And we know why it doesn't have a nucleus, because it is a prokaryote and not a eukaryotic cell. Chromosomes, animal and plant cells have chromosomes, right? We know why, because they have their DNA enclosed in a nucleus. Our bacterial cell does not have its DNA enclosed in a nucleus. What we saw in the bacterial cell was basically a nucleoid, free-flowing DNA, just like that. And then right here, we have the nuclear membrane, and animal cell, plant cell, and the nuclear membrane basically surrounds the nucleus. If it don't have a nucleus, it not gonna have no nuclear membrane. So we can understand why the bacterial cell is a no-no for that one as well. Then animal cells, we know our animal cells do not have a cell wall. They have an irregular shape, kind of roundish, irregular. They don't have a cell wall. Plant cells have a cell wall, and bacterial cells have a cell wall. Now, large permanent vacuole, only the plant cell has this, and we know why. The reason being that plants absorb a lot of water. That is why it needs such a huge vacuole in the center. For example, an animal cell processes way less water than a plant. Then chloroplast is only found in a plant cell, and we know why it's only found in a plant cell, because it's needed to carry out photosynthesis, and plants are also autotrophs, meaning that they're able to make their own food, versus an animal cell and a bacterial cell, which do not fall under that category. And then we have our plasmid, which basically we said it's like circular DNA that can only, so far we only see it in a bacterial cell, right? It's only in the bacterial cell, and basically it has the ability to carry genes. So we know that much about these three. Okay, so next up we're going to look at cell specialization. So basically, cell specialization is very important in multicellular organisms, right? So for example, a cell, for example, like our bacteria cell, which is very much unicellular, it's extremely small, so you know that it would have a high surface area to volume ratio, right? So something as small as a bacterial cell, which is extremely small, doesn't need cell specialization, because honestly it not really go out much. But look at our multicellular organism cells, right? If you choose to look at it from, look at, for example, the human body, right? We can choose to look at it from that perspective. Imagine if in the human body we only had just one type of cell that do everything. One type of cell are basically working our stomach to absorb nutrients. The same type of the one cell we depend on in our brain for we think and all that stuff, right? So imagine if that was the case. You think that would have worked out? Because as living organisms and as how big a human be, you think you would have worked out if you have like a one cell, I try to run your brain, I run your stomach, I run movement, um I control the muscles, all that. It would work out, it would work out at all. And that is why cell specialization is so important. So in our multicellular organisms like you and me, we need different cells to perform different functions, right? In order to obviously increase efficiency. Because imagine say if you sit down and you do only one thing at a time. You can only move your hand at a time, you can only talk one at a time. Like you cannot be talking and not moving at the same time, because yes, your cells them can't manage it, right? Imagine if that was the case. But cell specialization allows different cells to do different things, and obviously it increases efficiency. And we would kind of look at it from the perspective like it's a division of labor, right? So we're like a certain cell type focused on, okay, run the brain. For example, we have some neurons in our brain, I work on that. Then we have our blood cells in our bloodstream, I do what it's supposed to do. And yeah, so basically it's good to have different cells doing different things in order to increase efficiency and allow multicellular organisms to work properly, because if we have one cell that do everything, it would not make no sense. Then now we're going to look at transport in our cells, right? So basically there are three, actually let's go to four. There are four main types of transport across our cell. Diffusion, of course. Then we have facilitated diffusion, which kind of come off of diffusion. We have osmosis, and then we obviously have active transport, right? So basically all four of them very important and very necessary for the cell. So the first thing before we will talk about diffusion, right here, you need to be able to identify what this is. So this right here is our cell membrane. So this the word plasma membrane is the same thing as cell membrane. So what we have here is the cell membrane, and these are the heads and these are the tails. So so these are the heads and these are the tails. So the heads are obviously hydrophilic, these are hydrophilic heads, and then these are hydrophobic tails. That is why they're facing inside, meaning that they don't like water. So for the heads, they're hydrophilic, them like water, them like, all of that. And the tails, absolutely not. And that would be our membrane, right? That would be the membrane. So for diffusion, basically it's where substances are able to move across the membrane from an area of high concentration Obviously, we can see out here very much concentrated. So that's the high concentration to over here, which has a very low concentration until it's very much evenly distributed, right? So that's basically what diffusion is. Now, if you were talking about facilitated diffusion, that would work at all. The whole point of facilitated diffusion is that the cell needs help, right? So for example, there is a molecule, maybe glucose, and glucose can't come across this because glucose is not able to pass the hydrophobic tails, right? So in order to come across the membrane, it has to pass through this. So what's going to happen? We're going to need the help of protein carriers or carrier, yeah, protein carriers and no, protein channels and carrier protein. So we're going to need both of those in order to get across the cell membrane. So that's what we would use in the case of facilitated diffusion. Obviously right here there is no photo, but you get the point. There would be a protein channel which is kind of like a hot dog shaped thing, and it would help the particular molecule, for example maybe glucose, come across the membrane because it's not soluble, it can't pass the hydrophobic tails. So that would be a good example of facilitated diffusion. So then now we have osmosis. So basically this is where it is the movement of water molecules from an area of high concentration to an area of low concentration across a semi-permeable membrane until it is evenly distributed. Now, basically osmosis is very similar to diffusion, but the main difference is that it's taking place in water and it's also taking place across a semi-permeable membrane. That's what the main difference. So right here, we're going to leave the high concentration and go to the low concentration, right? So that's the whole point of it. So high concentration here to low concentration, right? So at the end of the day, the goal is always equilibrium. The goal of everything is always to become balanced. So for example, you see how high this side B is going to basically, alright, the way we consider it sometimes is we consider osmosis to be diffusion, but in water. It's kind of like the same thing, but except in water. So the goal is to become balanced somewhat. But that's basically the idea behind osmosis. So now we have active transport, and basically it's the movement of particles against a concentration gradient. Now this is the only, so basically when we talked about diffusion, facilitated diffusion, and also talked about osmosis, those three are all methods of passive transport, right? So them being passive transport means that they did not use any ATP. They don't need no energy in order to move across the membrane, none whatsoever. No, active transport is the only one that needs energy in order to move molecules across the membrane, and it's also the only one that moves particles against the concentration gradient, right? So that's the main difference between active transport and the other three methods of passive transport that we spoke about. The key thing is ATP, and it uses a channel also in order to move these substances. But also the next main thing about active transport, in diffusion osmosis, we said that we move from an area of high concentration to an area of low concentration. With active transport, it's actually the opposite. We're leaving from a low concentration to a high concentration. So there's basically like a flip when you talk about active transport, right? So ATP against a concentration gradient, and also we're moving from low to high concentration in the case of active transport. So that's probably one of the main differences between active transport versus passive transport, which would be the diffusion, facilitated diffusion, and osmosis. Now we're on the topic of nutrition. So basically, organisms need food to survive, right? And they need to convert this food into energy that the body can actually use. And basically this is done through nutrients, right? So we need nutrients in order to supply our bodies, right, with what it needs to grow, repair, and also regulate bodily processes. So our main types of nutrients are, obviously, we have our carbohydrates, proteins, fats, vitamins, minerals, and water, and obviously all of these would fall into the 6 food groups. At least when me that learn it, I did 6 food group. Whenever still 6 food group, don't have no, but me learn 6 food group. But basically our carbohydrates always provide energy, and even fats too provide energy. Proteins, they build and repair tissues. Vitamins, so vitamins and minerals are what we would call micronutrients. We need them in smaller amounts, and they basically help with body structure and function. So those are the 2 vitamins and minerals that would be considered micronutrients. We obviously need a lot of carbohydrates, a lot of protein, and somewhat lots of fats. Like, maybe the fats not really so necessary because the main thing is to provide energy, but we definitely need carbohydrates and proteins, and we obviously need water. Then we have the different types of nutrition. So earlier we did say that plant cells are autotrophs, right? They produce their own food. That's why they have the chloroplast, and that's why the chlorophyll within the chloroplast can absorb the energy from the sunlight, right? So autotrophs can make their own food. Example are plants or algae. And then we have us, which would fall under the category of heterotrophs, right? So we cannot make our own food. We depend on other organisms in order to make food. That's how we eat plants, that's how we eat vegetables, that's how we eat animals, that's how we eat chicken, all them stuff there. And then we have our saprotrophs. So basically those are the ones that feed on dead matter. It's like our fungus and our bacteria, and basically they kind of help to break down those stuff. For example, when something dead, you start to the bacteria upon you, start to the fungus just take over, that kind of situation. And then you have our parasitic nutrition. And when we talk about parasite, we know say some host involved, where one organism I go find a host, infect the host, just take over, and basically it will end up harming it at the end of the day. For example, tapeworms. And then we have symbiotic nutrition, where organisms live together and exchange nutrients for mutual benefits. So now we have different types of symbiotic relationships. We have mutualism, commensalism, and also parasitism, which we talk about parasitism right there. Then we could say commensalism is basically where one person or one organism benefits without harming the other one. So it kind of just did it and just get what it need, but it not really harm the other one. And then obviously mutualism, where both organisms are actually benefiting, like you do something for me do something for you. So it's a 50-50 situation in that case. Then we can talk about the process of photosynthesis in green plants really quickly. So we know that photosynthesis is the process by which green plants make their own food, right? We know that. And the thing about it, it's not just even plants, but you also have certain algae that's able to carry out photosynthesis like that. But at this level, let's just focus on the green plants. So if we're talking about photosynthesis, the number one thing is to remember the formula. Very important. And yeah, so obviously you're going to need carbon dioxide, you're going to need water, then you're going to need sunlight over chlorophyll, the same chlorophyll was in the chloroplast or in the plant cell, that same chlorophyll, and it's going to produce glucose and oxygen. So basically C6H12O6, that's the formula for glucose. And then oxygen is oxygen. And in some cases, Many of them will ask for balanced equation, but just know so you put a 6 before everything except the glucose. So our glucose is C6, H12O6, and then for the oxygen, just put a 6 in front of it. For the carbon dioxide, put a 6 in front of it. For the water, put a 6 in front of it. The whole balancing equation, that is getting down into some chemistry business, which we're not doing right now, but you just know so one 6 to go in front of everything but glucose. So now we know what the process is. We know that chlorophyll is 100% essential in order for photosynthesis to take place. Now, if all these factors are not present, there can be issues, or they can be less what should I say, less photosynthesis output. For example, less food would be made, or there's some limiting factor. So for example, we don't have enough CO2, but we don't have enough water, or we don't have enough glucose. It can be not glucose, but enough sunlight, or maybe the chlorophyll. We can definitely have issues when producing food in the plant. So we have two reactions. We have our light dependent and our light independent reactions. So obviously the light reaction is the light dependent reaction, and just a quick overview, it involves photosystems 1 and 2, photolysis of water, and basically that means splitting of water does take place and oxygen is produced. And then this occurs within the grana of the chloroplast, and basically the grana, so within our chloroplast there are some structures that are called the grana, and basically they are just stacks of like discs, and those discs are called thylakoids. And then in the end, for the light dependent reaction, ATP and NADPH are going to be produced. Then for our light independent reaction, aka the dark reaction, independent meaning you don't need light. No photosystem is necessarily required, and photolysis, meaning the splitting of water, does not take place. But in this light independent reaction, CO2 is absorbed. Now this occurs in the stroma, and basically the stroma is within the chloroplast and is basically the space within the chloroplast. So in the light dependent, we said it was the disc, which we said those are thylakoids, and a stack of them is called a grana. But for the light independent, we're saying that it happens in the stroma, which is the space around inside of the chloroplast. Right, so in the end, we're going to get reduced NADP for the light independent reaction. So now, earlier I said that some things can be limiting, right? So some stuff can limit the rate at which photosynthesis takes place, and it can limit the amount of food that plants also produce. So literally, if we follow the formula in which it said we need carbon dioxide, we need water, we need sunlight, we need chlorophyll present in order for photosynthesis to take place, right? For it to take place. So obviously light intensity, so the higher the light intensity, the faster the rate of photosynthesis up to a certain point. But too much light can actually damage the plant cells. And also for CO2, very important. Temperature, also very important, because if the temperature is too low, the plant can't go carry out photosynthesis. If it's too high, the plant still can carry out too much photosynthesis. And then if there is not enough water, the plant will literally struggle to carry out photosynthesis, and you can obviously see the plant start like wilting, which that would be a good condition. Then obviously you have chlorophyll concentration, so the amount of chlorophyll that we have in a plant affects its ability to absorb light, and then that influences the rate of photosynthesis. So let us take a look. So now we're going to look at some graphs and see what's going on here. So now three out of what we just mentioned now, very important, right? So basically the first graph right here, all of them say the same: rate of photosynthesis, light intensity. So obviously the more light, the faster the rate of photosynthesis, right? So what's going to happen is that the rate of photosynthesis is going to increase until something else becomes, obviously it's going to increase until something else becomes the limiting factor. So for example, we have the plant good, we give the plant enough light, we give it water, we give it carbon, target enough carbon dioxide, and then you max out the light, you max it out, and then something are going to become short, right? Something is going to become limiting, maybe the CO2 or something else, but something are going to become limiting and kind of flat out the rate. So basically at this point the rate of photosynthesis cannot get any higher, so it does balance like a plateau on the top of a mountain. It does flat, it does bottom, it just maintain that rate because the plant cannot photosynthesize any faster than that. And as I said before, if the light is too much, like it way too much, the plant might not respond so well to like that amount of like wavelength of light if it just way too much. But as I said before, something become the limiting factor, and what can happen is the plant just cannot photosynthesize any further. It reaches max and it just start level off. For the second graph, it's basically almost the same thing. We have enough carbon dioxide, we're supplying the plants with carbon dioxide, and the plant good, right? But the plant getting all this carbon dioxide, the plant cannot photosynthesize any faster. It get everything it needs, and it cannot. This is where it basically maxes out, and you see the flat off, kind of just like the light intensity. So at this point, something else is limiting the rate of photosynthesis, right? Something else will limit it. Maybe the plant short of water, maybe the temperature get higher, something. But even though the carbon dioxide are going with itself, something become I remember the principle, but something become the limiting factor, and something hold back the plant. Something is holding back the plant. Maybe the light gone down or something or whatever, but it just level off at this point because it cannot photosynthesize any faster or any more than it already is. It just flat off right there. So then for the temperature in the plants, yeah, so we know that we have a lot of enzymes involved in most of these processes. So what's going to happen is with temperature, it's going to start off, it's going to increase, right, reach the optimum temperature, because every organism has an optimum temperature that it works best at. So right here in the plants, it max out right here. So this is the optimum, right? So you can tell, say here, so the lower temperature did lower. So as you increase the temperature, go down, you see the rate of photosynthesis start off low, it build up, and then as you got closer to the optimum temperature, you see it reach a peak and then start flat out, right? And also, if you want to talk about temperature, we know that certain enzymes become denatured, right, at high temperatures. So what can happen is the temperature never optimum here, so right, but as you get closer to the optimum, it continues to increase, right? And then when it reaches the maximum, and then the high temperatures over here, so the temperatures get a lot higher, the enzyme protein denature start breakdown, and bam, the rate of photosynthesis start to drop, right? So that's basically how I would explain it. Then we have minerals. So basically, still on the topic of plants, and minerals are very crucial to plants. They absorb water and nutrients, with water and minerals through their roots, and there are three main ones, especially that they use commercially, and that's basically NPK, so our nitrogen, our phosphorus, and also our potassium. Very important. So our nitrogen is very important for the production of chlorophyll, and we know the function of chlorophyll. We need it to absorb light. And then if there is a deficiency of nitrogen in our plants, we can start to see some yellowing on the leaves basically, and then So some common signs of nutrient deficiency: stunted growth, the yellowing on the leaves, and necrosis. Then phosphorus, now we know that it's mainly involved, this mineral is mainly involved in energy transfer. So basically all that ATP and all that stuff, it helps the plant in the aspect of with energy. So basically also root development, flower seed production, and basically all of those things require a lot of energy in order to absorb the water from the root. And even when the seed and the embryo and all that stuff, that requires a lot of energy when everything is just being formed. So the effect on the leaves, that it improves overall plant vigor, supports strong leaves and all that stuff. So a deficiency in phosphorus can cause something like purple lining, like kind of like around the younger leaves. So that's how you would know if a plant is deficient in phosphorus. Right, so now common signs of the phosphorus deficiency: same stunted growth, leaves with purple lining, and then also poor root growth. Right, because we need strong roots in order, we need strong roots and we need energy in order to absorb all that water from the roots, right, from the soil. So now potassium, which is symbolized by K, so don't be fooled, phosphorus and potassium are two different something. Phosphorus is P, potassium is K. So the function of it, it helps with water regulation, right? So the effect on the leaves, it strengthens leaf tissues, promotes healthy leaf function, helps with stomata regulation, and a deficiency can lead to leaf edges turning yellow or brown and probably even curling up, kind of like, right? So common signs of it: yellowing, stunted growth, and low productivity within the plant. So now let's talk about the different tests. So within biology, you can test for different things, right? So we can test for protein, you can test for starch, we can also test for fat, and fat is another word for lipid in science, basically. So for protein, we can do the Burette test, and for example, if we did this on maybe egg whites, right? So what we're going to do, the procedure, just to jog your memory, we're just going to add a few drops of this Burette reagent, right? So that's how we would get the egg whites, put a few drops on it, kind of like mix it up or shake it up or whatever. And basically how we would know if there's protein present is if it turns purple. So with the Burette test, we expect it to turn purple, right? If it's negative and there is no protein present, we expect it to turn blue, right? So do not mix this up with the starch test, because we'll soon get to the starch test, right? But positive result for the Burette test is that it turns purple. A negative result, it turns blue. Now for the starch test, aka the iodine test, we can use iodine to test for it. So for example, maybe we can get a leaf, or something. Drop a couple drops of the iodine on it, right? So if we did the experiment with the leaf, we boil the leaf, we put alcohol, we cleanse out the coloring and all that stuff, and we did this. If the leaf turned blue-black, we know that there is starch in it. And if the leaf turned brown or reddish brown from the iodine, we know that there is no starch present. Now for the lipid test, that's the Sudan 3 test. So basically you would add a few drops of it to the sample, right? We dropped a few, add a few drops of it, and then what we would expect is that it would turn red orange, right? So basically we're looking out for red orange, that's the color we're looking out for, right? So if red orange pops up, we know that there is some fat or there's some lipid in it. If there is no fat in it, then we expect no color change, or we just expect it to just step back the same color as the Sudan 3, basically, right? So that those are three of the main tests that you can do: test for protein, test for starch, and also test for fat, right? So now enzymes. Alright, so now our enzymes, they speed up reactions, right? They are biological catalysts that speed up reactions. Now, each of them are highly specific, meaning that each enzyme typically catalyzes a single type of reaction. So it means that enzymes have different function. They're very specific. So this enzyme cannot work for that something. It's very specific, right? So each enzyme typically catalyzes a single type of reaction or works on a specific substrate, aka a reactant. So each of them have an active site that is very, very specific. So how they work, enzymes, they lower the activation energy required for a reaction, making the reaction occur faster and more efficient. Now basically what that means is, for example, let's just say that the activation energy, let's just say, just for example purposes, for a reaction, the activation energy, let's just say we need, not even like a use unit, let's just say in order for a reaction to take place, we need 20 something, right? We need 20 in order for it to be initiated. In this situation, you add an enzyme to it, the enzyme are gonna make that reaction happen at 10, so it make it happen a lot faster, right? So that's really how enzymes work. No, once you talk about temperature, we usually talk about enzymes because enzymes being proteins, they get denatured at high temperatures. So for example, when we looked at that temperature graph before, we saw that the rate of photosynthesis, it increased until it got to the optimum temperature and then it fell, just a drop, drop, drop. So basically what happened there is just the proteins becoming denatured at high temperatures, and then there's a decrease in the reaction afterwards, right? So enzymes, they interact with its specific substrate, and then they form an enzyme substrate complex, kind of like a lock and key business, very, very specific. Now we're going to move on to reproduction quickly. So now we know about reproduction, that it is the process by which new offsprings are produced. We have two main types, sexual reproduction and also asexual reproduction. We know that sexual reproduction, process by which male and female gametes fuse and fertilization takes place. Asexual reproduction, there is absolutely no fusion of gametes, no male and female, no fertilization. The offspring just come just like that. There is no fusion of anything. So now just a quick comparison. So sexual reproduction, we know we need both parents, fertilization has to take place, and fusion of gametes has to take place. Asexual reproduction, we don't need two parents, we just need one, right? And then offsprings produced in asexual reproduction are genetically identical to the parents. Now, sexual reproduction is going to give you what should I say? Genetically different children. So there's going to be variation in offsprings produced by sexual reproduction. For example, human beings, right? Fertilization takes place and somebody have a baby. If the person have 5 kids, all 5 of them are come out differently, and that's the variation we're talking about. Them could have similar little more, but them very much different. Even though they might similar, them very different. So that's what we talk about variation, and that's the kind of like diversity that we're talking about. In asexual reproduction, for example, a plant performs asexual reproduction and it have offsprings. All of the flowers them look dead stamp, like literally the same thing. So back to sexual reproduction, it can help reduce the spread of disease, can be used to increase food production, but the only disadvantage that we have is that it takes a longer time to produce offspring. So now, for example, a human being take 9 months to have a baby. A plant's asexual reproduction and bam bam, the pitney just born probably in a week or something like that, right? So it's very different. But here we go. So now asexual reproduction, we says it produces a larger number, and the main disadvantage is that there is disease. So for example, the parents of the disease, and then that plant do asexual reproduction and have offsprings, and all of them are go born with the same, all of them are go form with the same disease that the parent had, there's going to be an increased susceptibility to disease and all that stuff. So that's the main disadvantage with asexual reproduction. Alright, so now with the male reproductive system, so we're going to just start with the scrotum right here. So basically, it's to basically protect, it's to protect the testicle, and it also helps to regulate temperature. So that's the main function of it. And then leading off it right here, so that's the function of the scrotum, to protect the testicle and also regulate temperature. And then the testicle here is basically to produce sperm. So it's this one we're producing sperm and also producing testosterone. That's the function of the testicle. Then we have the epididymis, and basically this is where the mature sperm is where it's stored basically. So the mature sperm would be stored in the epididymis right here, and then for the vas deferens, this is basically the tube where the mature sperm from the epididymis would travel, right? It would travel from the epididymis to the urethra. Urethra is way around here, and basically the function of the urethra is just to release urine and semen. That's really the function of the urethra right there. So now right here what we have is the seminal vesicle, and basically they produce a fluid that helps to basically nourish the sperm. So it nourishes the sperm and contributes to the semen. So if we go from the top, we say that this protects, regulates temperature, testicle produces sperm and testosterone, epididymis. We said that basically stores the mature sperm, then the vas deferens, it's a tube where the mature sperm travels through, seminal vesicle nourishes the sperm, and then the urethra is where urine and semen would basically pass through, right? That's basically the main part, and so obviously the rectum that have to deal with other stuff, but we don't necessarily need to focus on the rectum. Yeah. So basically down here, this is the vagina, cervix, the uterus, and then we have the fallopian tube and the ovary. So basically the ovary is where eggs are produced basically, and also where hormones like estrogen and also progesterone are also produced right here in the ovary. So eggs and hormones are produced from right here. And then we have the fallopian tube, and basically what they do is to transport the egg from the ovary to the uterus. So basically the main process is for the egg to be implanted in the uterus. That is how somebody get pregnant basically, right? That is how somebody get pregnant, for the egg to travel from the ovary through the fallopian tube, implant itself on the uterus wall, then that's how somebody get pregnant. Now when that doesn't happen, the uterine lining will shed every month, right? So that's the biggest difference. So the uterine lining is going to shed, and then that's where the blood just bam 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 bam, and that's what passes out as menstruation. But if a female does get pregnant, then the egg will travel, fallopian tube, implants, and then bam, they're basically pregnant. Then the vagina is the opening, right, where obviously menstrual blood passes through, where childbirth, everything happens through here basically. And then we have our cervix, right? So the cervix is kind of like a wall at the back of the female reproductive system. Well, not the back, but you get the point. But it's kind of like a protective barrier, right? So it prevents, let's just say, harmful substances from passing this point. So it basically acts as a seal for the uterus, prevents infections, stuff like that. And I guess it also allows the sperm to enter, right? So that's basically the female reproductive system. Then we can just go through the menstrual cycle really quickly. So basically it's a monthly series of changes that prepares the body for pregnancy. It's the time between the first day of one period and the first day of the next. So the average is about 28 days, but it can vary from 21 to 35 days. So during the first few years, it may be irregular, and then it becomes more, I would say, predictable as a female would get older, right? So it can be light or heavy, painful or pain-free, long or short, and still be considered typical. So it would last between 3 to 7 days, right? So what happens during the menstrual cycle? So estrogen, those levels increase, causing the uterine lining to thicken. So it's going to get thickened. Then what's going to happen is ovulation. The egg is released from the ovary, moves into the fallopian tube. Progesterone levels increase after ovulation, while estrogen levels decrease. Menstruation. So basically, if the egg isn't fertilized, both hormone levels are going to decrease and the lining of the uterus is going to be shed. So basically what's happening is that the body itself is preparing to carry this child, and if the egg is not fertilized, all that is going to be passed out as basically blood. So then this is just a quick diagram of it showing that the wall thickens, it basically builds up, the egg doesn't get fertilized, so the walls begins to fall, it basically fall down. And then over time, as the egg is released, the body start build up by the wall again. And so basically that's what happens every single month. The wall is gonna build up. If it becomes fertilized, then it stays. If it doesn't become fertilized, because that thickening of the wall is really to support the egg, right? The growing embryo or whatever you want to call it. That's the whole point of it. If it's not fertilized, the wall is just going to be continue the lining, uterine lining, aka the wall, is just going to continue to build up, break down, build up, break down, build up, break down every single month if the egg doesn't become fertilized, right? So the wall falling down is basically representative of the menstrual blood. Then we have birth control. So what we have right here is basically different methods ranging from less effective to most effective. At the bottom, we have spermicides, fertility awareness, meaning that you'd kind of like track it. And then obviously you have withdrawal, and then, yeah, so those are basically the least effective on this list. Then you have in the 78 to 82% effective, you have the cervical cap, sponge, condoms, diaphragm, that's in a different category. And then on the 92 to 97%, we have the shot, pill. We also have the ring and the patch, and yeah, I think maybe the ring going to, I don't know how this go, but anyways, yeah. Then at the top, which is 99% effective, we have sterilization, and that's basically where somebody would get their tubes tied, then having the implant, and then getting the IUD, right? So I think the implant goes in the arm, and then the IUD, I don't remember how that go, but you get the point. So those are basically the most effective, and obviously abstinence is also the most effective. Okay, now the issues with abortion. So the main things with it is that there is ethical concerns, right? Obviously ethical concerns in a sense where some people view it as wrong, as unethical, it's a life. Some people hold that belief system. Then you have legal aspects where in some countries abortion is illegal and you can go to prison for having an abortion. Then you also have medical risk where you know that it basically there are risks, especially depending on how far along you are in the pregnancy. to get rid of the child. It does harm the body in some sense. And then we have social stigma, where people will judge you, look down, and say, oh, it kind of blends in with the ethical concerns. And there is judgment and also social stigma, and it might also impact future pregnancies. That's also something to take into consideration. Then we have the effects of smoking. So, smoking is very harmful, damages every organ in the body, increases the risk of cancer, heart disease, respiratory problems, and other health problems. So, your immune system also becomes weakened, skin aging kind of happens a lot more, reduces your fertility, and all that stuff. But even more than just smoking, you have second-hand smoke that might affect persons with probably asthma, bronchitis, different things. So second-hand persons who inhale the smoke are also at risk. So, anyways, this is the end of the CSEC Biology crash course. You can go ahead and subscribe and also follow on Instagram.